Hello and welcome to DNA Uncovered, Class 6 of AKA Science's Forensic Science Kit. Lots of fun in store for us today, so let's go ahead and get some prep started. Number 1, assemble the trait tree by matching the letters and hang it up where everyone can see it. Number 2, organize the beads. These beads will be representing the DNA base codes of A, G, T, and C, with green for A, pink for G, blue for T, and yellow for C. Make sure there are enough beads of each color in bowls for everyone to access them. Third, prepare the blood. Label one 9-ounce cup, donor 1, and one, donor 2. Then do the same for 5 2-ounce cups each. Fill both cups with water, add the powdered milk to donor 2, then use the red food coloring to dye them both red. And finally, fill the vials labeled anti-A with water, and the ones labeled anti-B and anti-RH with vinegar from the bottle in the supplemental bag. Quite a few vocab terms this time around. Number 1, blood type, a classification of blood based on the presence or absence of certain proteins. Blood can be A, B, AB, O, and either positive or negative. Therefore, there are a total of 8 types of blood. DNA, the material present in every living organism that contains all the genetic information about that organism. DNA sequence, the specific order in which bases and base pairs are arranged in a section of DNA. Donor, a person who provides something to another, like money to a charity or blood for a transfusion. Gene, a unit of DNA transferred from parent to child that confers a part or all of a trait. Hereditary, similar to inherited, something determined by genetic factors and able to be passed down through generations. And trait, a genetically determined characteristic, something encoded by DNA, like eye color. Oof, alright, let's move on to the trait tree. This activity is about testing for 3 genetic traits. PTC testing, that's the stuff that makes broccoli taste bitter, attached earlobes, and tongue rolling. In the case of yours truly, that would be no. Attached, yes. So pass out the trait tree leaves and the PTC strips and have your students mark off their traits. Then have your students follow the tree branches of their traits on the trait tree and stick their leaves on the appropriate area. Keeping those results in mind, we can dive straight into the second activity, DNA beading. DNA is made of 4 base nucleotides, which we have represented by green for A, pink for G, blue for T, and yellow for C. Pass out the beading the code worksheets and pipe cleaners and have the students slide the 5 bead codes for each of their traits onto the pipe cleaner. You're probably saying to yourself, that only looks like half of a DNA strand. Well, that's because it is only half of a DNA strand. Flip over the worksheet and tell your students that for DNA codes, A will always pair with T and G will always pair with C. Armed with that knowledge, have your students bead a second pipe cleaner with the opposite pairing and spiral them together for that beautiful double helix. Have your students compare their DNA sequences, and we can move on to blood typing. Begin by asking your students if they know their blood types. I have no idea what mine is and always forget to ask when I go to the doctor. Pass out to each student or group 2 sets of 3 vials, the blood typing worksheet, and a sample from each of the donor cups. Starting with donor 1, use a pipette to squirt some of the sample into the 3 vials, check for clumping, then check off the results on the worksheet. Repeat the process for donor 2 and compare the results to the chart on the worksheet to show that donor 1 has O negative blood while donor 2 appears to have B positive. Now clean up, debrief, and case closed. Thank you as always for joining me. Any more questions, check out akascience.org and meet me in the penultimate Class 7.